Assalamualaikum, dear students. We have discussed the three important steps of aerobic respiration, that is glycolysis, link reaction and Krebs cycle. Today we will take up the fourth and the final step of this aerobic respiration, which is known as terminal oxidation. This terminal oxidation, it is the final step of aerobic respiration, which involves release of protons and electrons from reduced coenzymes, that is NADH2 and FADH2, to oxygen. The protons combine with oxygen to produce water, that is known as the metabolic water. And in this step, which is known as the final step of this respiration, aerobic respiration, release of protons and electrons from reduced coenzymes, that is NADH2 and FADH2, which are produced during the first three steps of respiration, that is glycolysis, link reaction and Krebs cycle. They are released to oxygen. They are reduced coenzymes, and that is protons and electrons from reduced coenzymes to oxygen. These protons combine with oxygen to produce water, which we call as metabolic water. NADH2, NAD plus 2 protons plus 2 electrons, FADH2, FAD plus 2 protons plus 2 electrons. And these 2 protons, 2 electrons and half of oxygen react to produce the metabolic water, which is the product of this respiration. And this terminal oxidation, it consists of two processes. Terminal oxidation, it consists of two processes, that is two steps: electron transport and oxidative phosphorylation. And the first thing, that is electron transport, and then the oxidative phosphorylation. Now we will discuss these steps. First we will take electron transport. This electron transport is a group of coenzymes and cytochromes. It is a group of coenzymes, as we have seen in the electron transport of photosynthesis. It is a group of coenzymes and cytochromes that take part in the transport of electrons downhill with the loss of energy at each step. And electron transport is a group of coenzymes and cytochromes that take part in the transport of electrons downhill with the loss of energy at each step. The energy liberated during transport of electrons is used in transporting protons. And the energy liberated during the transport of electrons is used in transporting protons. This electron transport system, or respiratory electron transport, REC, respiratory electron transport or electron transport, is located in the inner mitochondrial membrane. This is located in the inner mitochondrial membrane. The individual membranes of the electron transport chain are called electron carriers. The individual members of the electron transport chain, they are called electron carriers, and they include flavin, FES protein, iron sulfur protein, quinones, most important is ubiquinone, and cytochromes, that is cytochrome B, C, C1, A and A3. And these are the various electron carriers which take part in electron transport chain or electron transport system of respiration or respiratory electron transport chain. Most of them are prosthetic groups of protons, and most of these electron carriers are prosthetic groups of protons. Quinones are highly mobile electron carriers. And out of these, quinones are highly mobile electron carriers. Electron transport chain consists of 4 molecular complexes. Electron transport chain in respiration consists of 4 molecular complexes and 3 sites for transfer of protons across the membrane. And this electron transport chain, it consists of 4 molecular complexes and 3 sites for the transfer of protons across the membrane. It takes part in the synthesis of ATP with the help of 5th molecular complex, and it takes part in the synthesis of ATP synthesis with the help of a 5th molecular complex, that is ATP synthetase. ATP synthetase. The 4 molecular complexes are NADH quinone reductase, that is also known as NADH dehydrogenase complex. Number 2, succinate quinone reductase complex. Number 3, quinone this hydroxy cytochrome C reductase complex, and cytochrome C oxidase complex. These are the 4 complexes. Number 1, NADH quinone reductase, NADH dehydrogenase complex, succinate quinone reductase complex, quinone hydrogen cytochrome C reductase complex, and cytochrome C oxidase complex. And these are the 4 complexes which take part in electron transport chain of respiration. The complex first. This complex first, NADH reductase complex. NADH plus H plus formed in the matrix donates 2 protons and 2 electrons to FMN, that is flavin mononucleotide, forming FMNH2. FMN extends from M side and the matrix side of the membrane, inner mitochondrial membrane, to its C side, that is cytoplasmic side or the outer side. It releases 2 protons into outer chamber and hands over the electrons to FES molecule. And NADH, the first complex formed in the matrix donates 2 protons and 2 electrons to FMN in a flavin mononucleotide, forming FMNH2. FMN extends from M side of membrane, that is matrix side or inner mitochondrial membrane, to its outer side, that is C side, cytoplasmic side. It releases 2 protons into outer chamber and hands over the electrons to FES molecule. Complex second, which is known as succinate quinone reductase. From FES, the electrons are picked up by coenzymes quinone, that is COQ, like ubiquinone. Quinone becomes receptive and picks up protons from the matrix to form QH2. The latter can also be formed directly from FADH2. QH2, being mobile, passes to C side of inner mitochondrial membrane. Here it discharges a proton pair into the outer chamber and donates its electrons to cytochrome Bf molecules. Now complex third, QH2 cytochrome C reductase complex. Cytochrome B hands over its electrons to FES and later to another quinone COQ. On receiving 2 electrons, quinone is able to pick up 2 protons from the M side of the membrane to join to form QH2. The latter shuttles to C side and liberates its protons to the outer chamber. Now the complex third, cytochrome C, complex fourth, cytochrome C oxidase complex. While shuttling protons to the outer chamber of the chamber, QH2 hands over its electrons to cytochrome C1. From here, the electrons flow to cytochrome C, cytochrome A, and cytochrome A3. The later 2 are collectively called as cytochrome oxidase. They pass the electrons to an atom of oxygen, making it reactive as O2 negative. The anionic oxygen combines with 2 protons, forming a molecule of metabolic water. it is seen that electron transport chain starting from NADH plus H is able to push 3 pairs of protons to the outer mitochondrial chamber. FADH, which enters the chain one step below, sends 2 pairs of protons to the outer chamber. Pushing of protons into outer mitochondrial chamber creates a proton gradient across the inner mitochondrial membrane. It is converted into ATP synthesizing machinery by ATPase of elementary particles observed, which forms the complex fifth. And in this complex fifth, this ATP synthesis takes place, which we call as oxidative phosphorylation, which has been explained by chemiosmotic synthesis. And this we will discuss in the next lecture. This is all for today. Thank you. Assalamu alaikum.